So my name is Ellen Winesapple, and I'm excited to introduce our next speaker, Dr. Rebecca Riggins. Dr. Riggins is an associate professor of oncology at the Georgetown Lombardi Comprehensive Cancer Center, where her research focuses on hormone receptor signaling in breast cancer. Today, she'll be discussing how high-resolution chromatin mapping technologies, like EpiCypher's CUT&RUN services, can provide new insights into this complex disease. With that, please join me in welcoming Dr. Riggins.
So thank you again for the invitation to present to the scientists and to EpiCypher. And today I want to share a little bit about some of our emerging data in this really exciting breast cancer space. Ellen kindly noted that, our interest is in hormone receptor signaling in breast cancer, and that's true. We have a particular interest in these sort of lesser studied side of hormone receptor positive breast cancer. And that's increasingly important as this disease increases in its frequency every year in terms of diagnosis and unfortunately death due to disease. And so the story specifically I'm going to talk about today is one that arose from some studies that we began not too long ago, looking at this concept of crosstalk between the estrogen receptor, or ER, and a receptor tyrosine kinase, HER2. And that crosstalk is actually happening in the nucleus in this particular type of breast cancer that we focus on called triple positive breast cancer, which I will define for you shortly. I always like to begin my talk by doing the acknowledgements first. So I really want to thank my fantastic team within the lab, both current and past. Folks highlighted in pink are people who contributed significantly to the work that I'm going to show you. Special shout outs to Stanley Tam, who's a talented graduate student in my lab, Dr. Shaima Banasi, who's a research assistant professor with the group, and Dr. Sandra Swain, who is my clinical collaborator here at Georgetown Lombardi. So triple positive breast cancer refers to the concept that this is a form of breast cancer that expresses all three receptors that are the targets for targeted therapy in the treatment of this disease. And by that, I mean the estrogen receptor, or ER, progesterone receptor, or PR, as well as the human epidermal growth factor receptor 2, or HER2. This so-called triple positive breast cancer subtype represents about 10% of new breast cancer cases that are diagnosed each year in the United States. Of note, this is the very same prevalence as the number of cases diagnosed each year of triple negative breast cancer, or TNBC, but we hear a lot less about triple positive breast cancer. If you look at those statistics another way, just over half, depending on the study, of HER2 positive disease is also hormone receptor positive. And why is this important to consider? So we are at an advantage, theoretically, when we think about the treatment of triple positive breast cancer, because we have two sets of molecular targets for therapies that have shown significant benefit in terms of disease-free and overall survival in the clinic. When we're thinking about targeting HER2, we're thinking about a suite of monoclonal antibodies, and of course now ADCs that are important for targeting receptor tyrosine kinases on the surface. We also have some of the newer agents, these tyrosine kinase inhibitors that can inhibit tyrosine kinase signaling through really well-known, well-established pathways in the cytosol. When it's time to inhibit the estrogen receptor, we again have a suite of molecules at our disposal. We have aromatase inhibitors, which today are the first-line standard of care for individuals with hormone receptor positive disease, increasingly even in the premenopausal status when we would include alongside that ovarian functional suppression. Other compounds that we have to modulate estrogen receptor signaling would be the selective estrogen receptor degraders or downregulators, such as fulvestrant, and increasingly some of the newer, really exciting orally available SERG, elacestrant being one of them. And some of the gold standards, which still have a place in clinical treatment depending on the context, selective estrogen receptor modulators, or tamoxifen. So we might think that because triple positive breast cancer has a plethora of targets that we have an easy task ahead of us. The reality is we don't. Some of this is more complicated than I have time to get into right now, but part of this sort of heterogeneity in how this disease is treated comes from our standard of care guidelines. So the American Society of Clinical Oncology, or ASCO's guidelines for the treatment of hormone receptor positive and HER2 positive disease gives individual physicians a lot of choice in how they treat this disease. And so you'll see the part that I've boxed in pink for if a patient's cancer is triple positive, The guidance is HER2-targeted therapy upfront in the neoadjuvant setting plus chemotherapy, or endocrine therapy plus anti-HER2 agents in select cases, or endocrine therapy alone in select cases. So there's a lot of ors in that statement. But what happens when we treat individuals with triple-positive breast cancer the way that most individuals with any type of HER2-positive breast cancer are treated, and that is with upfront neoadjuvant anti-HER2 therapy coupled with chemotherapy? The reality is, in these settings, HER2-targeted therapy is less effective in triple-positive breast cancer as compared to breast cancer that only amplifies and overexpresses HER2. This is a really nice summary diagram of a whole host of different clinical trials. You can see the drugs used in those trials along the left, as well as the acronyms for the trials. And when we look at both progression-free survival and overall survival, and we stratify that by the expression of hormone receptors, i.e. ER and PR, versus not, we see a lot more red color in those cases that correspond to hormone receptor positive disease. And that is a shorter survival, i.e. a higher hazard ratio. And so this is for, I will note, lots of different anti-HER2 agents. It's not just one class of anti-HER2 agents that perform suboptimally in triple positive breast cancer. So because my lab is interested in the hormone receptor side of things, one of our goals for this project was to define what are the estrogen receptor-driven mechanisms of HER2-targeted therapy resistance in triple-positive breast cancer. And one of our first questions was, well, what happens to the estrogen receptor upon treatment with anti-HER2 agents? And so we know quite well from the literature that the estrogen receptor is a transcription factor. It engages DNA, it turns on, and in some cases turns off the expression of genes associated with cell growth, cell proliferation, cell cycle progression, and survival. So we did a very simple experiment in a triple-positive breast cancer cell line called BT474. These were treated short-term with either control or a combination of monoclonal antibodies, trastuzumab and pertuzumab, which throughout this talk I'll abbreviate as TP, for a short period of time, 24 hours. We chose this combination because today this is still that upfront first-line standard of care anti-HER2 therapy that is used clinically. And so what we did, we included our trastuzumab, pertuzumab, and we also included one of those selective estrogen receptor downregulators or degraders or SERDs, fulvestrant, called ICI. And in this triple-positive breast cancer cell line, we looked by qPCR at the expression of 3 really classical estrogen receptor target genes, things that we would very well expect to be induced by estrogen, or in the case of this particular experimental design, suppressed by an antagonist of the estrogen receptor like ICI in the blue bars, which we see all the way across. What we noted quite strikingly, however, was that treatment with the anti-HER2 agent, trastuzumab, pertuzumab monoclonal antibody combination, in most cases led to significant induction of estrogen receptor target gene expression. Now, there could have been a very simple explanation for these data. There are many times, many contexts in which receptor tyrosine kinase signaling like HER2 and hormone receptor signaling like ER can counteract each other. So it's almost like a game of molecular whack-a-mole, in which case, if we're inhibiting the RTK or the HER2 signaling, it might make sense that we would induce estrogen receptor target gene expression. Except for the fact that we see this same behavior in a different triple-positive breast cancer cell line, MDA-MB361, which is completely resistant to all forms of anti-HER2 therapy. So this led us to believe that this was not simply as straightforward as you inhibit one side of the pathway and the other one reactivates to compensate, because these cells are completely insensitive to growth inhibition by anti-HER2 treatment, including trastuzumab or pertuzumab. Yet we see across the board, to varying degrees, induction of ER target genes. And so this is when, in the project, we spent some time with the classic literature and reminded ourselves that HER2 does not always act at the plasma membrane. It's been long established that there is a fraction of HER2 that can be trafficked to the nucleus, and that nuclear HER2 can be a transcriptional co-regulator. Studies have demonstrated that the nuclear form of HER2 can associate with the promoter of COX-2 and promote its expression in this context. while others have shown the nuclear HER2 can bind to ribosomal DNA in conjunction with RNA polymerase I in the nucleolus to promote ribosomal RNA synthesis. And excitingly for us, there was a study conducted in clinical samples that in the context of hormone receptor positive disease only, i.e. no HER2 amplification, having nuclear HER2 in the nucleus predicted for shorter disease-free survival, specifically in those patients who were treated with the selective estrogen receptor modulator tamoxifen. So we went back to our experimental models, the BT474 cell lines, and we asked firstly, does treatment with trastuzumab and pertuzumab cause relocalization of HER2 off the membrane and into the nucleus? And in fact, the answer is yes. We see within 3 hours TP-induced HER2 nuclear localization in triple-positive breast cancer models in this somewhat punctate fashion, quantified here on the right. Because we saw an induction of ER target gene expression and we can see presence or expression of HER2 within the nucleus, we actually asked what I think was probably the most important question of the entire study, which is, does trastuzumab, pertuzumab, or does anti-HER2 treatment, which causes HER2, some fraction of it, to enter into the nucleus, does that affect estrogen receptor recruitment to chromatin? And in fact, the answer is yes. By chromatin immunoprecipitation coupled with qPCR, again, at 3 really classic loci for these ER target genes, the progesterone receptor enhancer, the TFF1 or the PS2 promoter, as well as the FOXC1 enhancer, what we can see is that in response to short-term treatment with anti-HER2 monoclonal antibodies, we see robust recruitment of the estrogen receptor to these classical ER target sites. Importantly, this does not happen when you perform the same experiment in ER-positive cells that lack amplification of HER2. In this case, trastuzumab, pertuzumab has absolutely no effect on ER recruitment to chromatin, here or elsewhere. So what these initial studies had done for us in terms of defining ER-driven mechanisms of HER2-targeted therapy resistance in triple-positive breast cancer was demonstrate that this combination of monoclonal antibodies, trastuzumab, pertuzumab, can induce ER-target gene expression and recruitment to target genes. And this is concomitant with HER2 nuclear and chromatin localization. So this led us to ask the broader question of whether trastuzumab, pertuzumab treatment reprograms the ER cistrome as a whole within triple-positive breast cancer. So in this assay, we partnered with EpiCypher, and again, returning to one of our triple-positive breast cancer preclinical models, treated short-term with either vehicle or the combination of monoclonal antibodies, trastuzumab, pertuzumab, for 3 hours. We found that quite strikingly, trastuzumab, pertuzumab treatment radically expands the ER cistrome in this model system. We can see that under vehicle-treated conditions, we have some level of ER binding to DNA, but there is a massive expansion in terms of the number of ER peaks that are seen in the TP-treated condition. These peaks, or these gains of peaks, are really across various cistrome features with specific enrichment in introns, as well as distal and intergenic downstream regions, which is to be expected for a nuclear receptor like ER. We note that under vehicle-treated conditions, ER peaks are covering motifs where we would expect the estrogen receptor to be binding, chiefly the estrogen response element, as well as the thyroid hormone receptor beta motif. However, in anti-HER2 or TP-treated cells, we see a redistribution of ER away from these more classical motifs and to motifs associated with homeobox domain proteins, specifically CUT2 and glucosid motifs. When we ask by gene set enrichment analysis what kinds of genes might be expected to be regulated by these binding events, what we find is that trastuzumab, pertuzumab is recruiting ER to a series of target genes that are really essential to the Wnt, as well as the RAGE, the receptor for advanced glycation end product pathway genes in triple-positive breast cancer. Some examples being the kinase PIM1, as well as the NF-kappa-B PIK65 family member REL-A. And so we can see enrichment of not just Wnt and RAGE signaling, but also NOD-like receptor signaling and mitophagy in this analysis, whereby if you'd like to get a sense of how to read this graph, any peak to the right of 0 are events that are enriched in the TP-treated gene situation, as those to the left are enriched in vehicle. And so this is, interesting to us because there's been a really interesting push lately, both in the clinic and preclinically, to look at the role of these receptors for advanced glycation end products, or RAGE, in the context of where stress and our circumstantial environment actually affects outcomes of therapy. And that's because many of these receptors in this RAGE signaling pathway engages a lot of pro-survival, such as JAK-STAT and NF-kappa B signaling. So recall at the beginning, I made the statement that we've known for quite some time that regardless of what type of anti-HER2 therapy we use, it is less effective in terms of disease-free and overall survival in individuals who have tumors that are not just HER2 amplified, but also express the hormone receptors. And so we took these genes, particularly focused on those that regulate the Wnt and the RAGE pathway in these models, and we asked in clinical samples whether these might actually have any impact on survival outcomes. And strikingly, what we see is that high expression of this gene signature of Wnt and age-associated genes is associated with shorter distant metastasis-free survival in HER2-positive disease, but not in ER-positive disease, i.e. only hormone receptor positive. Moreover, while the sample numbers are low and we need additional ones to provide better support, there is the suggestion or a trend that high expression of these Wnt and age pathway genes engaged by the estrogen receptor are enriched in individuals who do not obtain a pathologic complete response in following neoadjuvant treatment with trastuzumab. And so here again, we've gone on to show that not only can TP or trastuzumab, pertuzumab induce ER target gene expression and ER binding at these sites, but that in fact, trastuzumab, pertuzumab expands the ER cistrome in triple-positive breast cancer to include Wnt and RAGE pathway genes, and that these gene products in clinical samples are associated with poor survival. So finally, we so far have asked whether trastuzumab, pertuzumab can more broadly influence epigenetic modifications in triple-positive breast cancer, again in partnership with EpiCypher to perform cut-and-run for histone modifications. The first of these that I'll show you data for is H3K4 trimethylation, where we see under baseline conditions enriched K4 trimethylation at BMEB motifs, and that there's a redistribution to 6-4 motifs upon treatment with TP. What we can see in TP-treated cells is a shift and a gain of H3K4 tri at genes associated with the serotonergic synapse, as well as protein processing in the endoplasmic reticulum, endocytosis, and the mTOR pathway. On the flip side, when we look at histone H3K27 trimethylation, we see a lot of this mark deposited at androgen receptor half-site motifs, which is something interesting that we're definitely going to want to come back to. And we also see sort of this global loss with a couple of notable exceptions in H3K27 trimethylation upon acute treatment with trastuzumab, pertuzumab. And so what I've shown you so far is that we have this really interesting outcome in that trastuzumab, pertuzumab, which is one of the classic ways in which we inhibit HER2 in HER2-positive disease, irrespective of hormone receptor expression, actually induces ER target gene expression and redistributes estrogen receptor DNA binding, not just to promoters and enhancers for some of its target genes, but more globally expands the ER systrome to include genes associated with poor survival in HER2-positive breast cancer. And finally, that this is accompanied by altered histone H3 methylation across a wide range of sites that may also be associated with poor outcome, but that will be the topic of future studies. Some of those ongoing and future studies are to actually validate at the gene and locus-specific level some of these TP-induced ER cut-and-run targets, specifically in this Wnt and RAGE pathway, as well as analyzing a brand new series of samples with which we work with EpiCypher to perform cut-and-run in primary and recurrent triple-positive breast cancer human tumor samples. And so with that, I would like to say thank you, and I would be happy to take any questions that you may have.
Fantastic. Thank you so much, Rebecca, and thank you, EpiCypher, for that wonderful presentation. Let's jump into our Q&A. As a reminder to the audience, you can feel free to submit your questions using the Q&A tab on the right side of your screen. To start us off, Rebecca, I was wondering if you have any data or if you're thinking about looking into how your results apply to HER2 low populations with this kind of increasing interest in using targeted anti-HER2 therapies in those spaces.
Yeah, no, I love this question. We are thinking about this. Many of the preclinical models that we might think about using to address this question, when we get into these studies, many of the models that we think about in terms of addressing this question really actually are HER2 low. And what do I mean by that? So the models that we rely on are truly HER2 amplified, but even some of the cell lines that we might, for example, consider to be hormone receptor positive only, they actually do have HER2, and depending on the situation or the culture conditions, you might actually cause them to express some HER2. Clinically, we have not moved in the direction of doing some of the validation in HER2 low, but now that that's an increasingly important population that's receiving certain types of anti-HER2 therapy, I think that's only going to become more important.
Absolutely. Thank you so much. Ellen, do you have any questions for Rebecca?
Yeah, so I'm not super experienced in breast cancer research, but I found the implications for Wnt signaling and the RAGE pathway very unexpected. So how would that impact drug research? Would people be looking for additional therapies that can target these pathways? Are there therapies that target the RAGE pathway?
Yes, there are. And in fact, that's really one of the near-term directions. There are a couple of clinically available RAGE inhibitors. One in particular, TTP-488, is a small molecule that has had a lot more attention in triple-negative breast cancer. But actually, one of sort of the next near-term goals would be to look at whether in this context, including one of those RAGE inhibitor small molecules might actually improve outcomes.
And I can follow up with another question.
Yeah, sure.
The changes in H3K27 methylation, the repressive mark at, I think it was androgen receptor.
Yes. Mm-hmm.
What do you think that could possibly mean?
So I think if I had to bet, my bet would be that is ultimately going to lead to enhancement or re-engagement of nuclear receptor signaling, potentially beyond the estrogen receptor. So there's a little bit of a fight in the breast cancer field in the hormone receptor positive only space, I will add, on the role of the androgen receptor and whether that is sort of something we should be inhibiting or something we should be activating. And so I think some of these models definitely have the androgen receptor. And so that's an area of follow-up to ascertain as to whether that is something that is getting re-engaged in response to trastuzumab, pertuzumab treatment. I wouldn't be surprised if it did.
Excellent.
Perfect. I think that might be the time that we have for questions. But thank you so much again for that discussion, Rebecca. And thank you, Ellen, for those great questions as well.
Thank you, everyone.